Alright guys, welcome to a skeletal muscle lecture. We're going to talk here about the structure and function of skeletal muscle only. We're not going to focus anything on smooth or cardiac muscle. That is for another class for you. But the majority is straight structure, and then we'll get into muscle contraction. And then we also talk about muscle fiber types and how those relate to endurance athletes. So to begin here, let's talk about the amount of muscles. You have about 600 plus muscles in the body. We are going to focus on some of the key important functions, and the first one being the majority of what we're going to focus on, and that's force generation as it relates to locomotion and breathing. These are the key components there to endurance athletes and what we do on a regular basis. Another force generation capacity is the postural support. This is essential to keep your body erect and throughout the spine and constraining the movement within or the degrees of freedom of the joints as well. And then the final one is heat production. This is done through shivering, but also during the muscle contraction, you actually produce heat as a byproduct of performing the work as well. Two very key components of skeletal muscle during locomotion and activity is that there's two functions as a flexor or an extensor. And those we'll get in. We won't touch too much on what the differences in flexors and extensors are. That's for your 245 class with origin insertions and what muscles are in particular. So for this class, the gross anatomy will not be a focus. But moving on to the basic structure of a muscle, and you won't be testing this, remember, but I think it's important that we review a little bit for those that may not recall what it is. But going down from A to the overall skeletal muscle, you break it up into the fascicle, and then you break it all down to the muscle fiber level. And you have these sheaths. The sheaths are fascia sheaths that wrap and support and bring everything together. And there's three layers that hold the sections of muscle together. And we'll go all the way down to the endomyosin, which wraps the groups of the myofibrils, or your muscle fibers, forming a muscle fiber. And then you have the paramecium wrapping groups of muscles, fibers together that form a fascicle. And then you have the epimyosin wrapping the groups of the fascicles, forming your overall skeletal muscle. The understanding of the skeletal muscle contraction focuses more on the muscle fiber level. This is where we go all the way down to C in the corner there, and talk about the individual myofiber within the sarcolemma and the sarcoplasm. The sarcolemma is another transparent tubule that kind of wraps around the muscle fiber, and it's considered the cell membrane. Now remember, we're not going to focus on the gross muscle, so we're going to go all the way down to this myofiber level and talk about it there. If you can talk about parallel fibers, parallel muscles and pinnate muscles, but again, that's for me a biomechanics class because that deals with the force generation capacity of the muscle. So here we're just going to talk about how muscles contract or the theories behind how muscles contract. So we'll break it up even further down, and we're going to go all the way down to that myofiber level, which you'll see inside the sarcolemma. So know that each of the muscle fibers contain numerous myofibrils. It's the individual myofibers that contain the sarcomeres, which contain the actual contractile components of the skeletal muscle. And for the purpose of this, we know that sarcomeres, as you can see in the line in the picture there, they run from Z line to Z line. So that is what you have as your one sarcomere. You can see the series of sarcomeres joining together that form your myofibril. Okay. So know parts of the muscle fiber that are going to help us in coming up here. And the myosin is known as the thick filaments. So you can see those in the red there. And the actin of the thin filaments, you see that in the gray or the blue if you look all the way down to the C portion of this graph. In combination, these two proteins give skeletal muscle their striated appearance. And so they are structural proteins, and so we know that. But other components here that we have, the A band is the thick portion of the sarcomere, which covers the myosin proteins. We have the I band. Those are the light regions of the sarcomere. These are only containing the actin part of, so there's no myosin within this portion. It's simply actin. But they're combined with other structural protein like acetinin, which attaches the actin to the Z disc. Now, acetinin is not something that we're going to have to really know too much about. And I said acetinin, it's actinin, and so it's basically just actin with an IN added to it. Don't worry about it. We're not going to go further into it. But then you have the H zone. This is only present with the myosin, so this area lacks the actin. So the areas that have the overlap, this is where a lot of the action goes, and you'll see the overlap either increases or decreases as we get to the structure, the contraction portion. Of course, it's going to increase when we get to the contraction, and then they decrease when we get to the eccentric contraction, because they become further and further apart. So to break it all down a little bit more, the structural proteins are the function that transmit the force generated by the sarcomere. So it is these proteins, and a few other the proteins that we're going to talk about, that are actually responsible for that force generation and transmitting it to the tendon. There are other few portions here that we need to discuss, and really kind of tie into muscle contraction. And we'll start with the mitochondria. The mitochondria are considered um, the powerhouse of the cell. They are a bag of they contain a bag of enzymes responsible for changing the energy contained in the food you eat into the molecules of adenosine triphosphate. So that's why they're considered the powerhouse, because they actually produce the energy that we use for muscular contraction. Then you have the nucleus up there on top. This provides the ability of a muscular growth due to the protein synthesis. And then over to the right there, we'll go to the transverse tubules, and these are those series of blue tubes that transfer the action potential from the sarcolemma and the sarcoplasmic reticulum. And then you have the sarcoplasmic reticulum, which contains the calcium storage sites. So this is where the storage of the calcium is, that is used to initiate and keep the muscle contractions in line and in process. Going further, this is a depiction of some of the additional structural proteins in addition to actin and myosin, because there's numerous of them thought to aid in that force production and the contraction of the skeletal muscle. And their role, again, is to transmit that force production to the adjacent tendons. Some of these are key to understanding that of one of the reasons why eccentric muscle contraction produces more force. It's thought of some of these portions going further. So to begin, you have the non-elastic titin. Titin is thought to bind myosin to the Z-disc, while this elastic portion over here, it's the elastic portion of the titin, containing the PEVK domain. Now P is proline, G glutamine, or E is glutamine, I don't know why, but V is valine and L is lysine. So again, this is the part that is thought to contribute to that passive force development during the eccentric contraction. So this is where you're able to produce a greater force during that eccentric contraction, because as you can see, that elastic portion, it's kind of wound up like a spring. So as that stretches, it's getting taut, like kind of like think about doing a plyometric. You're taught to store and release that stretching or that stress through force production, and that's what the PEVK domain is doing there. Then you also have this desmin. The desmin is bonds to adjacent sarcomeres to the Z disc. Again, it's not really fully known what its job is, but we just think it contributes to that. And then you also have another one that's not depicted here. It's nebulin. This attaches to the actin, and it's thought to regulate the number of actin and the length of the thin filament during the sarcomere assembly. So this is also thought to play some sort of role in the regulation of that actin and myosin interaction, especially through the number of binding sites that you see available. So let's go back to those key components that we talked about a little earlier. And this is kind of what it looks like if we took an image of what the mitochondria and certain things would look like. So again, key components, the mitochondria, as we said earlier, was a powerhouse. It produces the energy in the form of ATP. So it's the catalyst that converts that ingested food into that adenosine triphosphate, which again, through a series of pathways, which we will discuss later on, will convert into energy that we need. It's interesting. It's important to know that the mitochondria are free within the cytoplasm of a cell, and these are just a couple other images to show you cartoonish depictions of what it looks like, and then in the scan of a muscle. So another key component that's within the skeletal muscle are these fat droplets. You'll see it labeled TG there in the skeletal muscle to the left. But a fat droplet is essentially a triglyceride molecule, which is the storage form of fat in the body. And they are stored in many different ways in the body, and in a very key way is that within the skeletal muscle. Um, they are important for an endurance athlete, especially when they're stored in the skeletal muscle, because it removes the need to deliver the source of energy of the three fatty acids through blood, and it just removes it directly into the mitochondria of the skeletal muscle to form that energy, which is a great energy and time saver through that process for the muscular contraction. Um, but when we talk about it, let's talk about how we utilize this. So in the skeletal muscle, in order to use a fat droplet or a triglyceride for energy, you have to first break up the three fatty acids and the glycerol molecule. This is done through triglyceride lipase, but that's not important for this class. But just so you know, at this point, you have the three fatty acids and you have a glycerol. The three fatty acids go directly into the skeletal muscle where they are changed through the mitochondria and create the energy. The glycerol molecule, on the other hand, has to go into the liver and be created into new glucose units, and then it's going to be transferred back into the mitochondria. And this is through the process of gluconeogenesis there, and they are going to provide the energy that way. Now, it's important to understand that these provide a prolonged energy source where the amount of storage that can be within the skeletal muscles is much higher than your glycogen. So that is a very high source of energy, especially for your marathon runner. So the final key component we're going to talk about within the skeletal muscle is glycogen. Glycogen is the storage form both in the skeletal muscle and in the liver of carbohydrates, ingested carbohydrates. It's the second most important energy source inside the cell after the triglycerides. So this is very important, again, but there's a very limited storage form or storage capacity of glycogen, and truly kind of an unlimited concept of the triglycerides. So that's why we focus more on triglycerides. So now let's move on to skeletal muscle contraction. And this is going to be important for you to understand. This is one of the most important topics, or probably the most important that we'll discuss in this lecture. And this will probably be what your quiz question is on Friday when we get to class. And basically, it's just going to be discuss the series of interactions that occur to produce the skeletal muscle contraction. And it's going to begin with this excitation contraction coupling. And that is essentially the series of, as you can see, 8 steps here that result in that contraction of the skeletal muscle at the myofibril level. So the phenomenon refers to the sequence of events responsible for the muscle contraction, and it includes all the way the neuromuscular junction. So the nerve impulse arriving at that NMJ, the neuromuscular junction, and producing the release of calcium and everything. So we'll go through this series. Again, excitation is depolarization of the muscle fibers, and then you have the contraction, which is that muscle shortening and force production. And again, so the eccentric part of it is not follow the same pathway, but you do have remember that you do have some eccentric contraction as well. But we're going to deal with the shortening or the muscle shortening contraction concentric during this process. So the excitation process. So just a couple abbreviations that are on this slide that you need to know. NMJ is neuromuscular junction. The ACH is the acetylcholine neurotransmitter. It's a chemical that's released by the nerve cell to signal other cells. Okay, so this whole process of the excitation begins with the nerve impulse running down the axon and arriving to that motor neuron plate and then arriving at the neuromuscular junction. Okay, so this releases the acetylcholine into that synaptic cleft, so that cleft. And then the ACH binds, or the acetylcholine binds to the receptors on that motor nerve end plate down there. Now you can see it's series 1, 2, and 3. Okay. The end plate potential leads to the depolarization of the muscle. It travels, as you can see, down the T-tubule. and then the depolarization travels down that transverse tubule. Results in the release of calcium, and you can see the Ca2 plus, the calcium releasing into the channels that lead into the individual sarcomeres of the myofiber and the myofilaments. Further going in the contraction there for you, highlighted there in the red, the action potential reaches the sarcoplasmic reticulum, releases the calcium into the muscle, and then calcium is bound to troponin. We're going to go over more troponin, trypomyosin here in the next slide. But in the skeletal muscle, when it's absent of this action potential, it's important to know that the resting calcium levels are very low and very minimum, almost negligible. So the calcium does not influx the skeletal muscle and bind to troponin until that action potential causes the sarcoplasmic to release it. So the calcium is stored within the sarcoplasmic reticulum, awaiting on the action potential to release it into the skeletal muscle. Let's talk about the two T's, the T combo. This is a triggering step that it's regulated by two proteins. Those two proteins are the troponin and the trypomyosin, which are both held on the actin molecule. And these two guys are responsible for the regulation of contraction. Trypomyosin travels the length of the actin, which is the thin filament, and the troponin attaches directly on top of the trypomyosin at the locations of the active sites of myosin cross-bridge. So the troponin is that when the calcium attaches to it, it releases off the trypomyosin, exposing those binding sites that allow for the myosin head to attach and move. So the major theory that most people follow when they discuss muscle contraction is this siding filament theory. Now, I will say that remember that this is a theory. It hasn't been proven unequivocally yet. It's still the overall belief on how this behaves, but this series of events here discusses how that happens for you. So it suggests that myosin binding releases the adenosine diphosphate and a phosphate ion, and this is accomplished by myosin ATPase. The reaction creates the energy needed for a power stroke, and that power stroke is the flexing of the neck, bends to 45 from the normal cock position, then it forms that rigor complex fully contracting. This shortens the sarcomere during the contraction. Now the speed at which this happens and it occurs is dependent upon the activity of the predominant form of myosin ATPase. What that means is there are multiple forms. Some behave much quicker and some behave much slower, and this is one of those ideas of fast and slow muscle fiber type. Now, in order for this rigor complex, once that there's a fully contraction of the sarcomere to be released, a few things need to happen. The nerve impulse must be removed from the neuromuscular junction. Calcium is returned back to the sarcoplasmic reticulum from the troponin, which now causes trypomyosin to move back into its location, which blocks the binding sites on the actin. And finally, the mitochondria replenishes the ATP stores at the head of the myosin by the release of the adenosine diphosphate and the phosphate ion. So therefore, now it's replenishing that energy store to go further. So going back towards the Itzhal theory, remember that there's numerous cellular proteins and energy production systems that are involved in muscle contraction. But again, the most accepted theory is that of Dr. Huxley, and that's the sliding filament theory, in which the myosin creates that strong bond with an actin. So it comes up, attaches the myosin to the actin, and it does that power stroke. But once that power stroke is done, it releases and relaxes. And an alternative theory is that of Dr. Yanagata out of Japan, and he describes a tracking action of the myosin head along the actin filaments. So instead of going up and firmly attaching to a single actin, the myosin, more along the lines of tracks along by making little cocks, and you can see that on the left theory where it hits, moves on. These are loose couplings all the way up and down. And it's thought that this could be a possible explanation to what happens during eccentric contractions which is also a possibility. But a single power stroke of the myosin-actin interaction only shortens a muscle about 1% of its resting length. Either multiple contraction cycles need to be combined to reach the 60% of shortening some muscles, or this alternative theory of it sliding across the actin is a version that makes some sense as well. Um, so in the Huxley theory possible, um, it's a cock stroke attach release, and then that same myosin head would then reattach to a actin that's further than a line to reach that 60% shortening, or just a series of myosin create this looping down the line of the actin. The eccentric contraction, again, so this is not well understood, not fully understood, but the muscle sarcomere lengthens. Myosin heads are pulled out to 90 degrees, where before they were at 45 degrees, and it uses less oxygen and ATP, and that's the energy portion, than is a concentric. And again, this is thought due to that other force production per muscle there, um, units like the Titan, the Desmin, the PEVC, all those units are thought to aid in this because of force production per muscle fiber can be 2 times greater concentric than the eccentric. Um, but does eccentric activity increase or decrease injury? And that's, with this force production increase that happens, one of the theories is you have more muscle breakdown with eccentric activity than you do with concentric activity, and therefore that aids or increases your or injury production. So that's one thing you need to think about along the process. With all these passive units that are increasing that force production, you don't have the active compartments that are used to help decrease that injury rate. So now let's move on to the different muscle fiber types, and this is where some of the controversy comes, and um, ever changing. And I'm going to go through a very simplistic way to classify muscle fibers, but it's important to note that there are numerous ways, and it is thought today that instead of there being a simple classing of type 1, type 2A, and 2X like I have here, there's more of a continuum or a sliding scale in which there are numerous forms of these muscle fiber types within each fiber type classification we have. But classifications can be based on histochemical or biochemical characteristics of an individual fiber. Some of these include color, the number of mitochondria, and the speed of contraction. This goes back to that myosin ATPase that we talked about a couple slides previously, where the myosin, depending on what kind of ATPase it is, the contraction could be slower or faster, and that's where that comes from. But the main types that we're going to talk about here, of course, there are 2 main types, and then with one, there's a couple broken down. But it's slow, or type 1, could be called slow oxidative. Then there's the fast type 2, and these are 2A, which is a fast oxidative. And again, oxidative means there's still some oxygen involved there. And 2X, fast glycolytic, glyca, meaning that it's involved with and more of an anaerobic process of carbohydrate and glycogen. But one thing I want to make sure that we're pretty comfortable with and understand that a muscle is not one type, right? So the majority of your muscle is a mixture, but some are predominantly more type 1 and some are more type 2. And that's where you're saying that some people are more genetically inclined to be a sprinter versus an endurance runner and vice versa. But for the most part, there are a mixture within the skeletal muscles of slow and fast muscle fiber. But these are just a couple schematics. Again, characteristics of human muscle fiber types up there. Number of mitochondria, fatigue resistant, predominant energy system, ATPase activity. Vmax is the velocity max, so it's the speed of their shortening. The efficiency of it versus how cost effective it is to produce the energy. And then the specific tension, we can get over that a little bit later. But these are all how everything is classified there. You can look at it ranging from the fast fibers to the slow fibers. And remember the type 1 slow fibers, those are more of your aerobic fibers, more of what we're talking about and important for this class. But we will talk about later on some of the importance of of the fast fibers and more of that sprint training or speed training, and those come into play there. The immunohistochemical staining there, that just shows you if you use that method of classification, how the colors may differ. Again, that's just there for your knowledge, this slide here. Uh, so let's go further into some of the biochemical properties of skeletal muscle and fiber types. The oxidative capacity of a muscle fiber is determined by the number of mitochondria, and this is essentially the capacity of it to produce ATP through the different mechanisms. And then you also have the number of capillaries surrounding the fiber. The number of capillaries are important because this is a level of adequacy of delivering oxygen during contractile activity. If something has a higher number of capillaries, it has obviously a higher number of blood, a volume of blood that travels that may have a capacity to produce or to deliver more oxygen to the working skeletal muscle. And then with that, you have the amount of myoglobin within the fiber. Myoglobin improves delivery of oxygen from the capillary to the mitochondria because it acts as that binding site of the oxygen in the blood and shuttles the blood, the oxygen between the cell membrane and the mitochondria. So here you have you have the number of mitochondria, B, you have the number of capillaries, then you have the number of myoglobulin within the capillaries that shuttles back and forth between the cell membrane and the mitochondria. Um, this is very important in determining some of the fatigue resistance because if something has a high oxidative capacity, then it is thought to be fatigue resistant. For those things that have a low oxidative capacity, they are non-fatigue resistant and they succumb to fatigue quite rapidly, and that's what you get when you get your type 2 fibers is they don't have this high capacity, so they become fatigued much faster. They are only utilized in a short duration of time. But then you have the type of myosin isoform. And it's important to understand that there are numerous. I have three here, but there's numerous that are referred to in differing textbooks. So that way we haven't named any of them, but they determine the speed of contraction. What that means is the speed at which you break down ATP into that ADP plus PI, so from triphosphate into diphosphate plus a phosphate ion. The faster you can degrade the ATP into the ADP and PI, the faster you have the myosin head that comes up and attaches to the actin and producing that power stroke or the series of connections depending on which theory you believe happens. So that isoform is involved in the speed of the contraction. Um, so type 2 has a much faster isoform than a type 1, which has a much slower isoform. So then you have the abundance of the contractile proteins within the fiber. This refers to basically the number of actin and myosin that are comprised within the sarcomere. If you have a higher number of actin and myosin, then you can produce or a greater force production. So it comes in two ways, right? So not only the overall number, but actually, more importantly, the number that are involved in the contraction. The more that interactions you have between actin and myosin, the greater amount of force you're going to be able to produce. It's the size principle, and more going to if I'm lifting a 5-pound weight versus a 25-pound weight, right? Which one requires more actin and myosin to be engaged? Well, the 25-pound weight. So therefore, I'm producing more force. Sorry, I went a little fast there. All right, so now let's move on to the individual fiber types and some of the nomenclature we're going to use, and you'll see some other nomenclatures that I've posted there for you. But we're going to start with what we're going to, we'll come to know as those endurance type fibers, but they're slow or type 1 fibers. They're also known as slow oxidative or slow twitch. So this is where a bunch of nomenclature gets in the way and decide what you want to use for yourself. But for this class, we'll just call them slow type 1 fibers. They have a large capacity for aerobic metabolism, right? So this comes from their high mitochondrial content. And the reason why the mitochondria are so important because it's within this mitochondria that you have these enzymes that are so rich in oxidative power that can convert that ATP into energy quite easily and steadily and over a long period of time. They have a greater number of capillaries and myoglobulin concentrations than their type 2 counterparts.
And this is truly what gives them that red color, is that high myoglobulin concentration. So if you look at them under a microscope, and they'll slightly be red, depending on what kind of stain you use. And then we talk about the fatigue resistant concept. So this is that being able to use the oxygen for a long period of time and steadily work, because you're looking at that slower Vmax, or the slower contractile um, velocity, and that's due to the lower myosin and ATPase activity compared to those fast fibers as well. There's lower specific tension. Now this is due to a low number of myosin and actin, so therefore they have that less ability to generate a force, especially when we talk about the lower specific tension. If you remember from previous, it's per unit area of that cross-sectional area of the fiber. Um, so here, because they have less ability to generate force, even if it's the same unit area, they're not producing as much force. Um, they're more efficient than their type 2 counterparts, that's because they're using less ATP to produce the desired force. But again, your type 2 may be requiring a greater force output. So this is one of those interesting things. But if they were producing a similar force output, it would require less ATP. Now, it's important to note that depending on some training, and this is where it's kind of that gray area. You can't convert a type 1 into a type 2, but you can, and some athletes do have type 1 fibers that behave more like type 2 fibers. Right? So they can produce a higher force. They can um, contract more rapidly. They are a little more fatigable, but this is where it kind of behaves a little bit like the others. Um, Now it's important also to note that the contraction speed is not a cut and dry, and this comes back into that continuum and that spectrum, right? And that's because this ATPase activity is also on the spectrum, and it grades from fast to slow, not simply fast or slow. So this is why you don't have just type 1 and type 2. That's why you have a range within type 1. That's why you have a range within type 2. Um, so there are a couple subcategories of type 1 fibers. We just won't get into that very much because that's for a more advanced, just purely physiology course. Now we'll talk about these fast 2A fibers. So generally, these are also known as their fast oxidative glycolytic, and they're intermediate fibers. Intermediate because they behave a little bit like type 1. They go back and forth type 2. And this is the one where you're going to see it the most in your endurance runners or your endurance cross-country athletes. And actually, by that Salton 1977 paper, um, they call it the endurance athlete's fiber. They have more of this value within their system. It's about biochemical and fatigue characteristics between T1 and T2X. And so the biggest idea towards the type 1 fiber is that it has that high mitochondrial concentration. So therefore, it has the ability to process more ATP quickly and become more fatigue resistant. And this is greatest affected by training, right? So it's still a type 2 fiber. It's not going to become a type 1, but again, like you had type 1 that behaved kind of like type 2, this one is a type 2 that can behave a lot like a type 1, and you see this most in endurance athletes. So if a question comes up where I say, talk about muscle fibers and explain which one and why they would see this fiber more in endurance athletes, you would say type 2A fiber. You could say because through training, these fibers get an increased mitochondrial creation, so they take on that fatigue resistance and biochemical oxidative properties of a type 1, although they are still type 2. And then finally, the last one we'll talk about is the 2X fibers, or the fast fibers, also known as the fast glycolytic and fast twitch fibers. These are about 5 times more powerful than your type 1 fibers, but they have a small number of mitochondria, so this makes them very, very fatigable, right? So it's less resistance to fatigue, but it's also the greatest energy expenditure per unit of work, right? So these are very costly, non-economical type fibers. They are rich in their glycolytic enzymes, which is the large anaerobic capacity, right? So they are very, very rich in fibers that can turn over that carbohydrate and the glucose to produce energy. And then the fastest type of fiber in humans. Um, the highest Vmax is due to that highest myosin ATPase, right? And that so that's a contractile component there, the Vmax, and so this ATPase, you're going to find the fastest one there. Okay. So here's a couple more tables for you. The one on top is that one that we saw earlier, just a reiteration of what determines type 1, 2A, and 2X. Right, you see the 2A kind of goes back and forth, high to moderate, combination, high, high. It sits right in between the 2X and the 1s. You'll see the 1 is your aerobic style, and then the 2X is going to be your more anaerobic, more powered athlete type of thing. Table 2 again, here we go. So the percentage of slow fibers, type 1 in your distance runners, your track sprinters, and your on athletes. You can see in your distance versus your track sprinters that disparity, right? So track runners, sprinters, and distance runners, they kind of flip flop the type 1 and type 2 fibers there. It'd be good to see if it was split up into type 2X and 2A and how that would change. But your non athletes, no matter what, they're kind of right there in the middle. They kind of have a nice combination there of both right about the midline. And this is from the Tim Noakes book, so this goes a little bit more in depth on the physiological and physical requirements of sprinters and distance runners, which may explain a little bit why their muscle fiber types are the way they are. Think about the demands of each of the activities, and then what falls within these ranges, right? So that can help you think about it more sprinter and distance runner. Think about what those activities are. Sprinter, I'm thinking about 40-yard sprints, 100-yard sprints, 110 up to 200. Anything beyond 200 for me, you're starting to get more into the distance runner, depending on what level you're at, because of the time at which they're there. Olympic athletes of that 400 meter, I still can put that towards the sprinter aspect of it because they're still going fast. But even when you listen to the Olympics, they talk about it's a matter of who can slow down the least. Even the sprinters in the 100 meter, right? The reason Usain Bolt is so good is because he doesn't slow down, where the rest of them by the end of it are going to slow down because, again, the sprinters are so fast fatigable, and that's what's going on there. So again, the endurance athlete, the majority of fatigue resistance, slower contractile type 1. And you'll look down the line there. You can hang out explosive power, high versus very, very high. Aerobic capacity, for sprinters, they don't really care. Or distance runners, it's very important. And I would almost put that as critical or crucial. But again, we'll talk about that. So aerobic capacity, you're going to talk about your VO2 max, right? So that's going to be your capacity. But for me and what I hope to get through to you after this class is that the economy at which people are running at speed is more important than their capacity. Many people never hit their capacity. They can, at submaximal speeds, they can be pretty explosive. I mean, pretty cost effective and outperform some people that have a higher capacity. And then we'll talk a little bit about that muscularity thing, the disadvantages of or the perceived disadvantage of muscularity. It's muscularity is interesting when you talk to people about it. You don't have to be huge and be big and thick to be muscular. You can be nice and toned, but have some skeletal muscle that produces positive changes in your endurance, and especially from a injury perspective. So here's again just type muscle fiber, and this is split up a little bit more independently of activity, and this shows you what they classify. This is again out of Tim Noakes' books. The types of athletes: sprinters, jumpers, weightlifters, and then here we start to get into cyclists and swimmers. Now, cyclists and swimmers is one depending on the distance. You can be either a purely anaerobic athlete or you can be an endurance athlete. So that category there, I think they're lumping everybody into one, but that could also be split up like the endurance runners. If you saw the Olympics recently, you could see that those go hand in hand in two ways. The middle distance runners, there you are. So the 400 meters is where they start to the 1500 meters. And then you have half marathoners, right? So half marathoners only have about a 54% muscle fiber type 1. But then you get to those elite runners. Now, and again, these are elites, right? So this isn't your normal everyday mom or dad that's in a running club going out and running these distances. Um, but if these were elites, these are their composition. And sometimes, so that depends on how much you're actually training. Um, so that whole thing is this fiber type concentration determined genetically. If you come at a certain cultures, you're actually at a very young age put into an activity or sport um, based on body type and what they think you're best at. One of my old colleagues, he grew up um, at a very young age being forced to run half marathons and marathons. And he actually is if you ask him, he'll tell you he's a very aerobic individual. He's now one of the leading strength and conditioning coaches um, up in Oregon. And if you ask him, he's like, I should have never been running half marathons. I wasn't genetically determined to ride that. But I argue that anybody can run a marathon, and it's just a matter of picking the right speed and training properly. And you can convert some of this characteristics, again, like we talked about, to produce and benefit you. So just another little quick image that I got there for you. So you have your long distance slow twitch. You have your 400 to 800 meter, and that's that type 2A. Or for us, that's that one that goes back and forth. It's right at the border. And see here they use 2B. And for us, on our nomenclature, we had 2X. But that's that short sprinters. And it's funny that they use different characters. And look how the long distance runners, taller, more thin. And one of the things you'll look about at this is the direction of the body and how they're performing. The endurance runner's upright and he's heel striking. The middle distance runner's still heel striking with an overstride, but he's leaning a little more forward with the torso. And that sprinter's directly over the forward feet. These are all related to the cost of their motion and what makes them more economical at that speed and that style. All right, so to wrap up this with the force regulation of the whole muscle, so now we're going to go from the individual fiber to the entire skeletal muscle individual. So think about the biceps or think about your rectus femoris now. So we're summating all those smaller compartments and we're building up to the whole muscle unit itself. So the amount of force a skeletal muscle can generate is dependent upon a number of properties. And we're going to focus on three individual ones here. And generally speaking, it's related to the number and type of motor unit recruited, the length of the muscle, and then the nature of the neurostimulation within that motor unit itself, or within the motor units that make up that muscle. The number of motor units identifies the number of fibers that are contracting. So keep in mind the demand for force production. If the demand is low, you're only going to recruit a few motor units. If the demand gets higher, you're going to incorporate more motor units along the way, and therefore you're going to increase the number of muscle fibers that are being recruited and summating the force of that myosin-actin combination of force generation. But also in the number that you recruit, if there's a higher number of type 2 fibers, then the percentage is related to the percentage of increase in force production is related to the increase in the percentage of those type 2 fibers. So that's an important factor as well. Going into the length of a muscle fiber, think about a resting fiber. So if you take an image of that original, like any still drawing, and there's a limited overlap when it's resting of actin and myosin. So if they just completely go and attach there, the amount of force that's being generated there is minimal. And then if it's a fully contracted muscle, there's also the Z discs are so close that it doesn't have the ability to create a large amount of force. So when my bicep is my arm is as much as it goes up, I'm not producing the greatest amount of force because I'm not recruiting all the actin and myosin cross bridges to form that production. So there's some optimal length where I can have the greatest amount of interaction actin and myosin that are producing a large amount of force. This is easily measured in a dynamometer, or the isokinetic machine that we have in the lab, and you can see along a path where you can create the greatest amount of force. And you can do this anywhere you want, where you can hold the greatest amount of force at. So the final one is the nature of neurostimulation, and you'll probably know this a lot from twitches through the summation and then the tetanus. So as the neurostimulation and the temporal relationship of summated stimulations continues rapidly, eventually it will produce tetanus, where the stimulation goes and drops back into zero, goes up again, drops down to zero. Sooner or later, it's going to go so much that it doesn't drop down to zero, and you produce a greater amount of force that holds constant until fatigue happens and it's off again. But it's the amount, as the number of recruitments keep going, the time between relaxation decreases, and you're recruiting more and more and more and more fibers, and they summate together to produce that tetanus. So to sum it all up, for the class on Friday, what I want you to focus on is the excitation, contraction, coupling concept and process. That is going to be, I just want you to explain it in your own words. Again, what happens, nerve impulse comes down, it releases into the synaptic cleft. How, where, and when the calcium is released, where it goes, and how it attaches to which one, the troponin or the tryptomyosin, how that removes and opens up the binding site, and then the myosin head attaches. You can either do the sliding filament theory or you can do Unitas. It doesn't matter to me, whichever one you believe is the way it goes. You just have to explain the process, and if you want to explain the difference in the two, more power to you. But then also I want you to understand the differences in type 1, type 2, and type 2A and 2X. Now, I don't need you to go too specific. What I want you to just focus on is explaining which one is more prevalent for endurance, which one is more important for endurance, and which one you see a greater extent in endurance athletes and why that is. And then what I also want you to do is think about this concept of converting type 1 to type 2 or type 2 to type 1. And if you have time, see if you can find some literature on whether or not you can do it, because I think that's what we're going to focus on in class, and discussing that is a concept of muscle fiber typing, and whether through endurance activities you can change one to the other. And the same in strength training, and whether you can change one to another. And if these are the key determinant in performance. And then I also I'm going to show you some bonking, because I want to talk to you guys about that. That's going to set up some stuff in the future. Okay, so for the quiz, focus on the muscle fiber types and the excitation, contraction, coupling concept, and being able to explain those. Come with questions. I'm willing to go further into the length-tension relationships of a muscle and all those type of things. Anything you need me to go further with. So sorry this took me so long to get out there. And sorry this is a 50-minute presentation, but enjoy and come with questions. Have a good day.